my name is Fran Austriaco. I study crop sciences at the University of Illinois at Urbana-Champaign, and I'm currently a rising junior. This summer, I had the opportunity to work in the Seyfferth lab with Dr. Angelia Seyfferth as my research mentor. I helped collect data for the experimental rice paddies found at the University of Delaware. Various water and residue treatments are applied to each paddy so that we can monitor its effect on growth and methane emissions throughout the growing season. One method to measure this is by collecting pore water samples. We use a syringe to create a vacuum while it is attached to a rhizon. A rhizon is a rod that essentially has a ceramic tip, which can be used to collect water in between soil particles. So once it is attached to the syringe, we will stick it at an angle into the rice paddy soil and leave it there to collect the pore water. It takes a little bit depending on whether the soil is dry or not. Typically the drier ones take longer. And while we're out at the field, we also pipette the pore water into prepared reagents so that they're ready for colorimetric analysis back at the lab. We also take pH as well as ORP, or oxidation reduction potential, measurements of the pore water. Back in the lab, we take UV vis readings from these pore water samples to monitor the absorbance of ferrozene, sulfide, phosphate, and silicon in each paddy. In this clip, you can see me making some standards for ferrozene. Another measurement we're looking at is methane emissions because flooded rice paddies produce a significant amount of methane due to the microbes living in the oxygen-depleted waters. The way we measure this is by gas flexing. So we take a wheelbarrow which contains a gas net, which is the yellow device seen in this picture, as well as a little tablet that actually is collecting the data. We also attach fans under this tent-like structure, which we call a yurt, and then place it down gently over the rice paddy so that we're only taking the emissions from this specific paddy. And we also have to do it gently so that the soil doesn't get disturbed. We attach the tubes which connect from the gas net into the yurt, and then we start weeding. I just love finding snails and frogs just living their life in the paddies too. They're super cute. With the experience I gained from gas fluxing and pore water sampling, I began my summer research project. It focused on the effect of biochar on methane and arsenic levels in rice paddy soil, arsenic being toxic to humans and may be taken up by rice. I collected soil from the rice paddies which needed to be crushed and dried before I mixed it with the biochar. I used a drying rack, a mortar and pestle, and a sieve so that the biochar would be evenly distributed throughout the soil. It also lets me remove any rocks or roots that were collected while getting the paddy soil. These are the final bags of soil, which I then added to pots that had sand at the bottom. And the darker soil seen here is the one that was mixed with biochar, which is much darker than I thought it would be. Similarly to the field pore water testing, I inserted rhizons into the soil. However, these ones would be staying in there for the entirety of the experiment. These are also much smaller than the ones we used in the field. Here, I am sealing off the hole that was made to make sure the rhizon stays in place so that no water is leaking out. Once the plug and rhizons were installed, I added DI water to flood them so that it would mimic rice growing conditions. Now my pots are ready to be gas fluxed and collected from for pore water samples. At the time of recording this video, I had just begun this experiment, so I hope to present my results at the Summer Undergraduate Research Symposium.